All right, experiment 24 is about hydrolysis of proteins. So we talked about hydrolysis of polysaccharides last experiment, and this one's the hydrolysis of protein. So what a protein is it's composed of amino acids. So what an protein, so a whole bunch of amino acids together makes up a protein. So what an amino acid is it's got an amine group right here, so NH2, a carboxylic acid group, so C double bond O, bond OH. You might see that as COOH sometimes. It's got a central carbon right here. It's got a hydrogen coming off that central carbon, and it's got an R group of some sort. It could be various things coming off of that central carbon as well. So the central carbon's kind of got all four components to it. All the various different amino acids are right here. So these are all the different R groups, but if you look closely here, so let's take this one for example, you can see the NH2 group right here, the COOH group right here, and then there's an implied hydrogen coming off that central carbon, and then you have the R group of another carbon and an OH group. And that's what makes up serine right here. And so all of them, if you look at them, they all have that NH2, COOH, NH2, COOH. And they have something else coming off of that central carbon. With the exception of this one, but that's a weird, proline's a weird one. Anyway, so what you do, so these are all your amino acids. If you do a dehydration synthesis of two amino acids, you form a peptide bond. So what happens here is that a dehydration is when you take off of water, so you got an OH group coming from the carboxylic acid and an H coming from the amino group right here. And so when you take an OH and an H off, you get H2O, and you can link that C to that N right there. And that's what you see here. So you see C double bond O, and then that C is now bonded to that N. So you have C double bond O bond to that N. All right, and so that's going to be your peptide bond right here. So if you have a string of these amino acids that then fold however they will, that's how you get a protein. All right, so the string of the amino acids is typically referred to as a peptide, but then once they fold and get a tertiary structure, the primary structure, the tertiary structure, the secondary structure, and the tertiary structure, that's how you get a peptide like that. Or that's how you get a protein, is through that tertiary structure. So what proteases do is they are enzymes that hydrolyze the proteins into amino acids. So they do the opposite of that dehydration reaction in that they take this peptide bond and they add H2O to it to get two monomers of two amino acid monomers. And they are produced by microbes to recycle proteins. So if you ever see maybe a dead animal like on the road or whatever, there's probably some microbes growing on that and that are recycling those amino acids, so we can reuse those amino acids and so other things in life can reuse those amino acids. It's something that decomposers have, so to kind of regenerate the cycle, which was something similar to the carbon cycle that we talked about last video. This one is kind of like the cycling of amino acids, and that you can kind of use them or break them down or whatever it may be. Proteases are also in laundry detergent. Fun fact.
I thought that was interesting. They're in laundry detergent to break down your proteins, because we sweat proteins as humans, and so it's in laundry detergent to kind of break down your anything that makes your hose dirty. So I thought that was interesting because once they're broken down, they're very, very soluble in water, and so you can just kind of break them down like that. Now, one specific protein that we were talking about is called casein. You can actually buy casein as if you're a bodybuilder, pumping iron. You can buy casein as a protein that you eat or drink. You put them in a shake. I don't do this, but I have a friend who did this to build muscle, because it's a protein, so it has a lot of amino acids. So it's a very large protein present in, but it's also present in milk. So you can also just drink milk if you want. But it's present in milk, and it's what makes the milk white. And so this is a picture of your casein protein right here. So you see it's very, very large. There's some other fun facts that you don't have to worry about, but this is just a picture of it. And there's an enzyme called caseinase that specifically breaks down this big protein right here. And it's a type of protease that breaks down casein into those amino acid monomers. And it is an extracellular protein, so it gets secreted out of the cell. And that's kind of a general trend and extracellular enzymes we'll see have
pattern to them. So what we did with the caseinase experiment is that we had a milk agar plate like this, and we divided it right down the middle in two, and we streaked bacteria on this side and bacteria on this side. This side we had E. coli, this side had Bacillus subtilis. That's one of my favorite microbes to say, Bacillus subtilis. If the bacteria creates caseinase, then you can see it's going to be secreted, and it's going to break down that protein that makes milk white. So you get this clearing around. It's really interesting. You get this clearing around like the plate like this. While on this side, there's no caseinase produced because E. coli is our bitch bacteria. There's no caseinase produced, so there's no zone of clearing, and so the plate's still white. And if you remember, this is an extracellular enzyme. And if you remember, all the enzymes from last experiment were in extracellular enzymes as well. And we also saw a zone of clearing in that video. So if you notice that extracellular enzymes that are secreted can create these kind of zone of clearings because they're like the colony's going over here, but they can secrete them all the way out to here, right? And so secreted things, you can see a zone of hydrolysis a lot of time if they break down stuff. So that's an interesting pattern to note. But so caseinase is produced by Bacillus subtilis. Other bacteria can grow on the plate that do not produce caseinase and will have no zone of inhibition, right? So Bacillus subtilis has that caseinase enzyme. Another thing we talked about was gelatin. So we talked about casein first, and now we're talking about gelatin. So gelatin is a protein made by animals in connective tissues, and it's in bones. It also makes up Jello, which was a big craze back in the good old days when refrigeration became a thing. You used to like make all this Jello stuff. But certain bacteria can actually break down this gelatin, and it'll like take like something like Jello like this, and it'll like break it down into just being like a pool of liquid. So yeah, gelatin's a really interesting protein. But we can put it inside plates. So we took a plate that had gelatin dissolved into it, and we divided it in two, just like the caseinase experiment. We divided it in two, and we put E. coli on one side and Bacillus subtilis on the other. So gelatin is clear, so we can't really see it naturally. So milk is white, and we can see it. So when it's broken down, we know it's a zone of inhibition. But we cannot see gelatin naturally. So what we have to do is we have to put HCl in it. HCl will make the gelatin that's dissolved in the plate, it'll make it precipitate out of solution. And once it's precipitated out of solution, so it forms like a solid, once it forms that solid, it makes like a murky white kind of a clearing. So precipitate form is white, and if the bacteria is, so if you see a clearing around where they were so if the bacteria secretes gelatinase, then there will be no zone of hydrolysis because there's no precipitate in that like area right there because it produced gelatinase, right? So it broke down all the gelatin, so it didn't have the chance to precipitate. So again, we have Bacillus subtilis being able to break down this protein while so it has gelatinase in it, while E. coli does not have gelatinase. Again, it's the bitch bacteria. So the big takeaway here is that proteins are made of amino acids. It's important to know what an amino acids, but these proteins can then do a hydrolysis, so it cuts with water, right? So a hydrolysis reaction that can break down proteins and into those amino acids. And caseinase and gelatinases are both proteases that do this, and they break down casein and gelatin into their amino acids. So you can test for this by identifying a zone of hydrolysis around it, whether it be in milk or whether it be in a precipitated gelatin thing. But again, the major theme here is that testing for these enzymes can provide insight into what their genetics have and what their physiology is. So it's really important that you kind of get down that all these are physiology experiments that we're doing in the second semester. And I'll probably have that line in every single video I'll do. So again, this is just another example of me accidentally saying I have more to say, but then I'm really done with the video. So cool.